Secrets of a Sex-Changing Fish revealed. We may take it for granted that the sex of an animal is established at birth and doesn't change. However, about 500 species of fish change sex in adulthood, often in response to environmental cues. How these fish change sex has, until now, been a mystery. The secrets of fish that change sex have, for the first time, been revealed by an international collaboration led by New Zealand scientists and including La Trobe University geneticist and Prime Minister's Prize for Science winner 2017, Professor Jenny Graves. The findings were published today in the journal, Science Advances. I've followed the bluehead wrasse for years because sex change is so quick and is triggered by a visual cue, Professor Graves said. How sex can reverse so spectacularly has been a mystery for decades. The genes haven't changed, so it must be the signals that turn them off and on. Bluehead wrasses live in groups, on coral reefs of the Caribbean. A dominant male, with a blue head, protects a harem of yellow females. If the male is removed, the biggest female becomes male, in just 10 days. She changes her behavior in minutes, her color in hours. Her ovary becomes a testis and by 10 days it is making sperm. Using the latest genetic approaches, high-throughput RNA sequencing and epigenetic analyses, the researchers discovered when and how specific genes are turned off and on in the brain and gonads so that sex change can occur. The study is important for understanding how genes get turned off and on during development in all animals, including humans, and how the environment can influence this process. We found that sex change involves a complete genetic rewiring of the gonad, Dr. Erica Todd from the University of Otago, the co-lead author, said. Genes needed to maintain the ovary are first turned off, and then a new genetic pathway is steadily turned on to promote testis formation. Co-lead author PhD candidate Oscar Ortega Recalde, also from the University of Otago, said the amazing transformation also appears possible through changes in cellular memory. Chemical markers on DNA control gene expression and to help cells remember their specific function in the body. Our study is important because it shows that sex change involves profound changes in these chemical marks, Mr. Ortega Recalde said. La Trobe's Professor Jenny Graves said the project links to studies of sex reversal in Australian dragon lizards that she is collaborating on with researchers at the University of Canberra. With dragon lizards, the trigger for sex change is temperature, which overrides genes on the male sex chromosomes and causes embryos to develop as females, Professor Graves said. Sex reversal in dragons and the wrasse involve some of the same genes, so I think we are looking at an ancient system for environmental control of gene activity. HIV eliminated from the genomes of living animals in a major collaborative effort. Researchers at the Lewis Katz School of Medicine at Temple University and the University of Nebraska Medical Center (UNMC) have for the first time eliminated replication-competent HIV-1 DNA, the virus responsible for AIDS, from the genomes of living animals. The study, reported online July 2 in the journal Nature Communications, marks a critical step toward the development of a possible cure for human HIV infection. Our study shows that treatment to suppress HIV replication and gene editing therapy, when given sequentially, can eliminate HIV from cells and organs of infected animals, said Kamel Khalili, Ph.D., Laura H. Carnell Professor and Chair of the Department of Neuroscience, Director of the Center for Neurovirology, and Director of the Comprehensive NeuroAIDS Center at the Lewis Katz School of Medicine at Temple University, LKSOM. Dr. Khalili and Howard Gendelman, M.D., Margaret R. Larson Professor of Infectious Diseases and Internal Medicine, Chair of the Department of Pharmacology and Experimental Neuroscience and Director of the Center for Neurodegenerative Diseases at UNMC, were senior investigators on the new study. This achievement could not have been possible without an extraordinary team effort that included virologists, immunologists, molecular biologists, pharmacologists, and pharmaceutical experts, Dr. Gendelman said. Only by pooling our resources together were we able to make this groundbreaking discovery. Current HIV treatment focuses on the use of antiretroviral therapy, ART. ART suppresses HIV replication but does not eliminate the virus from the body. Therefore, ART is not a cure for HIV, and it requires lifelong use. If it is stopped, HIV rebounds, renewing replication and fueling the development of AIDS. HIV rebound is directly attributed to the ability of the virus to integrate its DNA sequence into the genomes of cells of the immune system, where it lies dormant and beyond the reach of antiretroviral drugs. In previous work, Dr. Khalili's team used CRISPR-Cas9 technology to develop a novel gene editing and gene therapy delivery system aimed at removing HIV DNA from genomes harboring the virus. In rats and mice, they showed that the gene editing system could effectively excise large fragments of HIV DNA from infected cells, significantly impacting viral gene expression. Similar to ART, however, gene editing cannot completely eliminate HIV on its own. For the new study, Dr. Khalili and colleagues combined their gene editing system with a recently developed therapeutic strategy known as long-acting slow effective release (LASER ART). LASER ART was co-developed by Dr. Gendelman and Benson Edagwa, Ph.D., Assistant Professor of Pharmacology at UNMC. LASER ART targets viral sanctuaries and maintains HIV replication at low levels for extended periods of time, reducing the frequency of ART administration. The long-lasting medications were made possible by pharmacological changes in the chemical structure of the antiretroviral drugs. The modified drug was packaged into nanocrystals, which readily distribute to tissues where HIV is likely to be lying dormant. From there, the nanocrystals, stored within cells for weeks, slowly release the drug. According to Dr. Khalili, we wanted to see whether LASER ART could suppress HIV replication long enough for CRISPR-Cas9 to completely rid cells of viral DNA. To test their idea, the researchers used mice engineered to produce human T cells susceptible to HIV infection, permitting long-term viral infection and ART-induced latency. Once infection was established, mice were treated with LASER ART and subsequently with CRISPR-Cas9. At the end of the treatment period, mice were examined for viral load. Analyses revealed complete elimination of HIV DNA in about one-third of HIV-infected mice. The big message of this work is that it takes both CRISPR-Cas9 and virus suppression through a method such as LASER ART, administered together, to produce a cure for HIV infection, Dr. Khalili said. We now have a clear path to move ahead to trials in non-human primates and possibly clinical trials in human patients within the year.